Now imagine that this bacteria is now coated with immunoglobulin G. The next step is eating of this bacteria, which is known as phagocytosis, and this was given by a scientist which is known as Elie Mechnikov. Elie Mechnikov is a scientist who gave the phagocytosis, and we have 3 steps in phagocytosis. How this bacteria is eaten up? The first one is recognition and attachment. First, the neutrophil or macrophage need to identify that this is the bacteria I need to eat. So first is recognition and then attachment. Then there is engulfment, that is putting this bacteria inside in the mouth. Suppose if you stays in a hostel or in a PG or somewhere and you order food, first of all you open the Zomato app and you need to see that which is the restaurant from which you are going to make the order. So you recognize the restaurant that you are going to make the order. Then you make the order, that is attachment. The next step is engulfment. The order has come, now you need to put food in the mouth. Putting food in the mouth is engulfment. Then the last step is killing, eating of that food. So recognition and attachment. The neutrophil or a macrophage has got a receptor for immunoglobulin G. Remember, this bacteria is coated by immunoglobulin G, that is opsonization, making the bacteria tastier. We have a neutrophil over here or a macrophage. This has got receptor for immunoglobulin G. It comes and attaches to the immunoglobulin G, and that is known as recognition and attachment. This neutrophil or macrophage has got a receptor for immunoglobulin G or mannose or scavenger receptor. Now that it has attached to it, its now purpose is to eat. How it eats is, first of all, there is formation of the pseudopodia. If say suppose this is a bacteria, first of all, this macrophage is going to make the pseudopods. It is going to take the hands. Pseudopods are like both the hands. It is going to take the bacteria, take it inside the mouth. So these pseudopods are like the hands, pseudopods of the macrophages. These pseudopodia are like a hand. They are going to engulf the bacteria, taking the bacteria inside with the covering of a membrane, and that is known as phagosome. If this is a bacteria, there is a membrane, and that is known as phagosome. When it takes bacteria inside with the help of pseudopodia, this bacteria is taken up by a macrophage with the help of the hand that are known as pseudopodia. It takes inside. When it goes inside, it is lined by a membrane, and this is known as phagosome. Then it fuses with lysosome. If this is a lysosome, it fuses with lysosome, and it forms phagolysosome. It forms a phagolysosome. You can say phagosome plus lysosome. This is phagolysosome. So this product is known as phagolysosome. If we zoom at this, the third step is going to be killing of this bacteria. Now zoom this membrane, phagolysosomal membrane. How this bacteria is killed. Now let's talk about the last cellular event, that is killing. Inside the phagocytosis, we have the last step, that is killing. Killing can either occur with the help of oxygen, that is oxygen dependent, or without the help of oxygen, that is oxygen independent. Now let's talk about killing with the help of oxygen dependent, which is the most potent, most common, which is also known as hydrogen peroxide MPO halide system. We'll talk why we are calling this such a big name. Let's take this membrane of the phagolysosome. If this is a phagolysosomal membrane, the most important enzyme over here we have is NADPH oxidase. NADPH oxidase is the most important enzyme. NADPH oxidase is the most important enzyme for phagocytosis. See, oxygen comes in a phagolysosome. This is a phagolysosomal membrane. When the oxygen comes in with the help of NADPH oxidase, now this enzyme NADPH oxidase, very important question is, take pH from here, take OX from here, and it is made up of FOX gene. NADPH oxidase is made up of FOX gene, pH from NADPH, OX from oxidase. With the oxygen dependent killing, oxygen we are taking, that is why oxygen dependent killing. Why oxygen dependent? Because with the help of oxygen, now we are killing the bacteria. How we are killing the bacteria with the help of oxygen? What we have studied earlier, that this oxygen is going to make a superoxide. which is a free radical. With the help of the superoxide, it is converted to hydrogen peroxide with the help of superoxide dismutase enzyme. This hydrogen peroxide is going to get converted to HOCl with the help of myeloperoxidase enzyme. HOCl is nothing but hypochlorous acid, or basically a bleaching powder. So inside the cell, our body is making bleaching powder just to kill the bacteria, and this kills the bacteria. This kills the bacteria. This is oxygen dependent killing. Because with the help of oxygen, hydrogen peroxide is formed. And from this hydrogen peroxide, we are using this myeloperoxidase enzyme. That is why we call it hydrogen peroxide MPO halide system. This is oxygen dependent killing. Another path is mediated by the arginine. With the help of the most common one we have already discussed, that is hydrogen peroxide H2O2 MPO halide system. Another oxygen dependent killing can occur via another path, that is mediator arginine, and with the help of nitric oxide synthase, it forms the nitrous oxide. This nitrous oxide is actually reacts with superoxide and forms the peroxynitrite, this one, and this is going to kill the bacteria. So peroxynitrite, it kills the bacteria. So basically this is with the help of arginine, another pathway. But the most important pathway that you need to remember is hydrogen peroxide MPO halide system. This is oxygen dependent killing. Another one is oxygen independent killing. Without the help of oxygen, you can kill the bacteria with the help of a lysosome, with the help of lactoferrin, with the help of defensin. And the most important one, that is major basic protein, MBP, major basic protein, is released by eosinophils. And we know that eosinophils are the one that are used to fight the parasitic infection. And we have also cathelicidin. Cathelicidin is the one actually with the help of vitamin D3. This cathelicidin is produced, and it helps us to fight against tuberculosis. So if a person is having vitamin D3 deficiency, that means the patient will not have cathelicidin. Cathelicidins are absent in that individual, and these people have got high risk of having tuberculosis, because this cathelicidin actually help us to fight against the tuberculosis.